All right, welcome everyone. So today we're talking about systems genetics. We're going to talk about heritability, polygenicity, and pleiotropy. And these are the awesome folks who help prepare the slides. So this is the last lecture of module 4. So we've talked about aligning and modeling genomes, gene expression, epigenomics, regulatory genomics and networks. We've talked about population genetics, so genetic variation more broadly. Then we've talked about genetic variation in the context of phenotypic variation. Then we talked about intermediate phenotypes and EQTLs, expression quantitative loci and mediation analysis. And today we're talking about heritability and systems level genetics. And then next time we're switching to comparative genomics and evolution, followed by the quiz. So the goal today is to go beyond the single locus view, to actually start thinking about the entire genome simultaneously. And that's where heritability comes in. So we're going to talk about heritability, its definition and key concepts, how to partition phenotypic variants and genotypic variants, how to estimate variants, and look at narrow sense versus broad sense heritability. Then we're going to talk about the genetic architecture of complex traits, about polygenic risk scores and linear mixed models, and how to partition heritability. And we're going to cover an omnigenic model of complex traits. And then we're going to talk about genetic architectures and systems biology and how to think about rank-based enrichments, genes, pathways, and regulators. And then we're also going to talk about phenotype predictions, how to impute intermediate phenotypes and also large-scale models and inference algorithms. So the key concept today is heritability of complex traits. So what are some of the lessons that we've learned from GWAS that are motivating today's lecture? The first lesson is that we haven't found all causal loci, that basically the known loci explain relatively little of the phenotypic variance. The second is that most loci affect transcriptional regulation. They don't tag coding variation. And of course, the biggest and most important lesson of GWAS is that it actually works, despite what one could have thought about looking at correlations between individual SNPs that are thought to, that are now recognized to have tiny little effects, all the way to disease phenotypes. Despite this very, very large path, there are 120,000 loci that have genome-wide significance. And yet, these capture a tiny little fraction of overall heritability, overall, genetically explained phenotypic variation. So what we're going to be focusing on is, how do we reconcile the two? So first, we're going to define the concept of heritability, which is very, very helpful broadly. And then we're going to look at polygenic risk scores, disease architectures, linear mixed models, how to partition heritability, a very important technique for estimating and partitioning heritability called LD score regression. We're going to talk about that. And then models of polygenicity and a recently described Omnigenic model of disease. So let's dive with heritability. What is heritability? So let's think about components of variation. Just one second. Let's think about the phenotypic, the components of phenotypic variation. So we're going to basically assume that the phenotype P is a function of your genetics, G, and your environment, E. And that the variation in P, the variation in phenotype, is related to your variation in genetics and variation in environment between individuals, and also the covariance between G and E, but which we're now going to ignore. So we're going to assume that there are no gene environment interactions, that basically, your environment does not interact with your genetics. Okay. There are cases where this would be the case, but for now, we're going to assume that perhaps after correcting for population stratification, the covariance of G by E is actually negligible. So that basically means that we can think of the phenotypic variation in a given sample as a component of the variance that is explained by genetics and the variance that is explained by environment. So basically part of your phenotypic variation is driven by genetics, and part of your phenotypic variation is driven by environment. Everybody with me so far? Lovely. Good. So now here's one example where your entire genome is driven by one genetic variant. Okay, basically this is the entire genetic contribution. There are 3 possible genetic values in the population, you know, having 0 alleles of the alternate, 1 allele of the alternate, and 2 alleles of the alternate. And the intuition is that V of G is the variance of the mean phenotype across genetic values. And V of V is the variance of the phenotype for the same genetic value. So, you know, from the QTL lecture, you should be used to seeing this plot where if I have, you know, homozygous reference and homozygous alternate, I have dramatically different means. And now the remaining variation, the residual of the variation, is in fact the environmental variation. So basically the G, the V of G, you can think of as the difference in means, and the V of E, you can think of as the residual variation. Okay, so who's with me so far? So we had 66, 33 in the previous one, and then 0, 0, 0. Okay, lovely. So this is the key concept here, that basically if I measure my phenotype with more noise, then the genetic heritability actually goes down, right? There's 78, 10, 0, 10, 0. So you know, this is a very powerful concept to embrace, the fact that if I measure my phenotype extremely precisely, then I can actually reduce the fraction of the phenotypic variance that I would attribute to the environment. And if I measure my phenotype less precisely, it will look like the environment has more of an effect, but that environment might simply be that, you know, the measurement error. Okay, so that's why heritability as this fraction, basically heritability is defined as the fraction of the phenotypic variance that is explained by genetics. And that heritability is dependent on how well I can factor out all of the other environmental components. And therefore, if I can explain away more of that variation, given that the genotype is fixed, then I can explain more of the, I can basically result in a higher fraction of heritability. So heritability actually depends on our ability to measure this variation, okay? So when we're building these components now of the genetic variation, V of genetics, we now can partition that further into different components. We can basically start thinking of the genetic variation between individuals being driven by, in part, the additive effects. So V of G is the portion of the phenotypic variation explained by genetics, can be explained by the portion of phenotypic variation explained by additive effects in my sample, by dominant effects in my sample, and also by interaction effects between the genetic variants, okay? So the additive component is effectively a linear model. It basically says, how well can I add up all of the linear effects of all of the you know, genetic loci that contribute in a linear model, and then that gives me V of A. Whereas the dominance component requires, you know, more complex models, and the interaction component is simply intractable for most applications because we simply don't have enough samples to estimate 6 million squared, basically, as potential contributors. But there are, you know, network-driven approaches that you could use to get at some of those interactions. And that's what, for example, Remy in my group is doing with CombiG. He's basically looking for combination and interaction effects driven by the network models. But this additive component corresponds to a linear model, and we can add more causal variants, and the phenotype becomes closer to Gaussian. So the more causal variants we add, the more Gaussian the distribution will look. And again, we could further decompose the interactions. We could include the variants due to de novo mutations, due to, you know, all kinds of additional contributors. So now we can think of heritability as that ratio of variances. So basically, if the phenotypic variance is a combination of both genetic and environmental variance, and that genetic variance can be further broken up into additive, dominant, and interaction effects, then we can talk about broad sense heritability, which is capturing the entire V of G, everything that can be explained with genetics, and what fraction of that V of G, what fraction of the phenotypic variance is in fact captured by the genetic variance, okay? So heritability is simply the fraction of V phenotype that is explained by V genetics, everything added up together. So that's broad sense heritability. Narrow sense heritability only captures the additive effects. And it basically says, what fraction of the total phenotypic variance can I explain based on additive genetic effects? And there's, of course, an ongoing debate about the relative importance of additive versus dominant versus interaction versus other effects in the human population. Okay, so who's with me so far on these concepts of heritability, phenotypic variation, genetic variation, the component of phenotypic variation that is driven by genetic variation, and then how we can partition the phenotypic component that can be explained by genetics by the portion that can be explained by additive, by dominant, or by interaction effects. Lovely. Okay, so we have 50, 40, 0, 1, 0, 10 here. So, of course, the big question is, why do we care about heritability? Because it allows us to quantify the importance of genetics versus environment in a particular trait of interest. It can teach us about genetic architecture, namely how many causal variants are there? What are their effect size distributions? What are their allele frequencies? And also narrow sense heritability is a fundamental parameter that's needed for phenotype prediction. And it's the theoretical best possible prediction performance with a linear model. So there's many reasons for studying heritability, especially as we start getting to a mature GWAS field, where we often ask the question of, are we done? Should we be looking for more genetic loci? You know, how far down the rabbit hole do we go? Are we missing something fundamentally different than the kinds of stuff that we found already? So you can estimate heritability in relatives by basically asking, what is the fraction of phenotypic variation that I share versus how much genetic variation do I share? Okay, so the intuition is that the heritability that relates phenotypic correlation to genotypic correlation. So I can basically ask across a set of individuals, how much phenotypic correlation do I see? How shared are the phenotypes versus how shared are the genotypes? So if two individuals have the same allele at every one of the causal variants, they will have the same phenotype. And at least they will have the same genetic contribution of the phenotypes. And then the residual is, of course, phenotypic variation that is attributable to environmental variation. So the Haseman-Elston regression basically says we're going to fit a linear regression of phenotypic correlations against genotypic correlations. And we're going to look for the slope between these GG correlations and these PP correlations between genotype pairs and phenotype pairs. And we're going to derive the genotypic correlation from family relationships. So monozygotic twins are going to share 100% of their genome. Siblings are going to share 50% of their genome, etc. And using this approach of basically looking at the similarity in height between twins, between first-degree relatives, between second-degree relatives, etc., we can basically draw a regression between how similar are their heights, and then infer that height should have a heritability of 73%, whereby 73% of the phenotypic variation would be explained by this genotypic variation, that basically this broad sense heritability or this narrow sense heritability, if I only assume an additive model, would be in fact driven by this relationship between the genetic variation and the phenotypic variation. Everybody with me so far? Yep. Awesome. So now, how do we estimate heritability from genome-wide association studies? So what have we done so far? We've basically looked at the effect sizes beta at every causal location in the genome and use that to predict the genotypically driven variation, okay? So we're gonna have basically a set of effect sizes multiplied by the, on top of the SNPs basically, which we're gonna use to predict the genotypic component of phenotypic variation. So we can estimate the SNP effects and we can estimate their effect sizes beta from genome-wide association studies. So the variance explained by every SNP depends on its effect size and its minor allele frequency. So if we take this minor allele frequency, F, 1 minus F, and then beta squared. So the variance that I can explain from this additive linear model of all of my SNPs is going to be driven by this equation, which basically tells me how much is every SNP contributing to the overall effect. And if we do this with genome-wide significant SNPs, what we end up with is a phenotypic variation that I can explain from GWAS being a tiny fraction of the total additive variation and the total phenotypic variation. But again, the additive variation that I could have predicted is in fact still greater than the subset of SNPs that are genome-wide significant. And the reason for this is just discovery power. We're simply underpowered to discover the full set of SNPs that are responsible for a particular trait. So for height, with 250,000 samples and 700 genome-wide significant loci, and you can think of them as ordered in a way that the strongest effect sizes are first and the weakest effect sizes are last, or at least the product of the effect size and the minor allele frequency, because some SNPs will be so rare where even their effect size is big, it will explain a tiny fraction of the heritability. So even if you sort these SNPs by heritability explained, the one at the bottom here, the 700th genome-wide significant locus, is only explaining, a tiny fraction of a millimeter in height. And, between the moment you wake up in the morning and the moment you go to bed at night, your height actually varies statistically much more than 1 millimeter. So you basically have genetic variants that are so small, and the remaining variants, are even smaller in the heritability that they explain. And yet, when you put all of GWAS together, you're basically at 16% versus what you should be explaining of 73%. Is everybody with me on this discrepancy? So the big question has been, well, something's got to give. Either I have incorrectly estimated the total contribution that genetics could get me, or I can, maybe I've miscalculated this heritability. Maybe this equation doesn't hold. Maybe GWAS is capturing a tiny fraction. Maybe there are there are effects beyond additive, such as dominant effects or interaction effects. Maybe there are combinatorial effects. Maybe there are all kinds of other effects that are breaking this model, or there are thousands upon thousands of additional loci remaining to be discovered. Okay, so, or there are rare variants of enormous effects that are in fact hidden within the haplotypes that are associated with your phenotypic variation, with your GWAS study, and that these variants, in fact, explaining an enormous amount of variation, but they're improperly tagged by the common variants within there. And that's sort of one of the sources of this discrepancy. So this has been known basically as the missing heritability problem. The fact that there should be some additional heritability that we're capturing, the 73% heritability between relatives that we can estimate. And yet GWAS only captures a small fraction of that. So there must be something else. And this debate is now sort of tilting on the side of, no, really, these are continuing to be effects of tiny little, weak effect SNPs, which are, together contributing to this residual phenotypic variation, but using this almost omnigenic model that we're going to talk about at the end. Okay. But of course, there are still proponents of and there are still examples of strong effect variants that are explaining some of that. There's still an open search for combinatorial effects that we've been largely underpowered to find, and there might be additional components. Okay. So who's with me so far on this whole sort of missing heritability problem, sort of understanding that there should be more explained than we're currently able to explain? Right, so we have 50, 30, 880. Okay, so that's about heritability. Now we can also ask, well, great, we might not be able to explain everything, but can we use GWAS variants and can we use all variants in the genome to try to predict the phenotype of a person using polygenic risk scores? So basically that means poly means many, and then genic, many genetic loci, and then risk scores predicting the risk of a person for a particular trait. So you can estimate the absolute risk combining both genetic and environmental risk factors. So you can basically say, your absolute risk is some, in influencing many clinical decisions, such as whether you should have a particular lifestyle, whether you should be screening for particular diseases, whether you should be making changes, whether you should be, getting counseling and specific preventative therapies and risk-reducing surgeries and so on and so forth. So when you're here, there's no harm. Basically, everybody should eat healthier and exercise more. But when you're here and you're asking, well, should I do some chemo prevention? Should I do some endocrine therapy based on my genetic risk? That's when you're getting into, murky waters of, well, what is the harm and how confident am I about the risk? So you can basically, distinguish individuals based on a lifetime absolute risk and, distinguish high risk, medium risk, or low risk individuals, and then combine genetic variation as a way to do that. The way that this has been traditionally done is through largely monogenic disorders or oligogenic disorders, where there's a very small number of variants that are contributing to this. But then the question is, can we now do this using polygenic risk scores, using many, many SNPs in the genome? So how do we estimate these polygenic risk scores? So using this univariate genome-wide association study statistics, we basically have the betas, which are the effect sizes, which are basically the log odds ratios of a particular SNP. And then we have a set of genotype dosages for 0, 1, or 2 alternate alleles for every locus. So what we want to do is predict the overall risk of a person by combining many, many variants. So that's what a polygenic risk score is, or PRS. So the polygenic risk score is the sum for all SNPs of the effect size of individual variants and the number of variants that I carry at every one of those positions, the number of alternate alleles. And the question is, can we just combine all the SNPs? Yes or no? So if the correlation is, between G and G2, if, between any pair of variants, the correlation is 0, then we can use an additive model. The other question is, can we trust the estimate beta of all of the SNPs? Perhaps for genome-wide significant SNPs, we can trust these estimates beta, but as soon as I get to extremely, low p-values, then these betas might actually not be, very good estimates at all. And then the question is, can we simply restrict ourselves to the genome-wide significant SNPs? Well, the challenge there is that the genome-wide significant SNPs capture a very, very small fraction of the total heritability, so they might actually not be the best estimate. So one common practice for PRS estimation is to basically simply use the sum over a subset of SNPs. And then we can tune the threshold of how to choose that subset of SNPs. We can basically ask, maybe we should be thresholding up to some p-value. Maybe we'll go to 10 to the minus 8. Maybe we'll go to 10 to the minus 4. Maybe 10 to the minus 2, which starts really getting to the murky zone. But surprisingly, there's still signal at such low levels. Also, should we be using all of the genome-wide significant SNPs? For example, in this locus, there are 8 different SNPs that are all associated. Are they independent of each other? And one way to overcome this is to only take the top one and then prune away all the remaining ones. But of course, all the remaining ones might have some residual information, even if they're not genome-wide significant after correcting for the first one, but there might still have some residual. So using all of them might not be good because it might overinflate the estimate. Using only one of them might be very bad because we're missing out a lot of remaining information. And we're going to look at ways to actually capture those dependencies by factorizing that entire matrix and sort of capturing the independent components of all of these SNPs. So we can basically also cross-validate with the observed phenotype. We can basically say, how well can I predict it using a held out set of individuals or using, additional, ways of evaluating the data. So to know whether the observed SNPs are good, we can basically ask, how well can I explain my heritability, my phenotypic variation, explained by genetic variation, using all of GWAS, using only the top 500 or the top 200 or the top 60,000 SNPs, and then ask, how is my area under the receiver operating characteristic, which is this sensitivity versus 1 minus specificity curve? How is that area changing as I include more and more and more SNPs? And as I include all of GWAS, I'm getting to 71%. Can I include more beyond just, all of GWAS? And are my estimates getting simply too noisy after that? So there's a challenge, of course, with using all of the SNPs. And the reason is that there's linkage disequilibrium between them. So basically, we will be adding additional spurious effects because some of that effect might already be explained by the other SNPs in that locus. So one way to do that is to do pruning and basically remove the additional correlated SNPs. Another way is to actually explicitly decorrelate the LD structure. So the idea is that if there are combinations of SNPs in that region that are together adding more information than simply every SNP in isolation, then you could basically look for the principal components of that space. And instead of thinking about individual SNPs and individual genotypes, transform this into a decorrelated eigenlocus space, where basically the independent effects are effectively captured in these. And the covariance is basically the identity matrix, and there are no interactions between these independent components. So basically, instead of having the original estimated effects, I now have a set of transformed estimated effects. And then that covariance is now that identity matrix. And therefore, I can actually think of each of these independent components. And those independent components are, in fact, combinations of multiple SNPs. But these combinations are, in fact, orthogonal combinations to each other. So we can transform the SNP space into a multi-SNP space using singular value decomposition. We can select independent and orthogonal factors, or we can regularize the eigenvalues to smooth out spurious correlations. And after this regularization, we don't need that much additional tuning. So there's some references by Shamil Sonyev's lab that basically is using this approach. So then the question is, how well are these polygenic risk scores capturing the heritability of different disorders? And if you take everything with, some p-value of GWAS that's down to 10 to the minus 4 or 10 to the minus 3 or 10 to the minus 2, or even just, something ridiculous as like 5 times 10 to the minus 2 uncorrected, and so on and so forth, you can ask then, what is the total, heritability that I can capture? What is the total predictive power that I can capture with these sub-threshold GWAS SNPs? And the aggregate burden of these subthreshold SNPs can actually improve prediction performance. As you include more and more SNPs in the risk score, the association with rheumatoid arthritis, with celiac disease, with myocardial infarction, and with coronary artery disease, in fact, gets stronger. And in practice, this requires tuning, of course, of the p-value threshold and LD pruning threshold and so on and so forth. But what's really remarkable is that the genome-wide significance threshold is 10 to minus 8. Here we are 4 orders of magnitude less significant than genome-wide significance. And we're capturing, only so much. But as you continue including more and more SNPs, you continue capturing a huge amount of additional heritability. So what this is basically telling us is that these very weakly associated SNPs are also playing an effect. And in fact, that there likely is an enormous number of additional contributors. So who's with me so far on this sort of polygenic risk score estimation, on this decorrelation of the SNPs within these loci to basically look for the independent effects, the orthogonal effects, and these effects are multi-SNP effects. And also the fact that as you include more and more SNPs, you continue actually increasing your predictive value. So we're at 33, 44, 22, 0, 0. All right, so we talked about heritability, we talked about polygenic risk scores. Let's now see what all of these are teaching us about disease architecture. So the first thing that we're going to do is actually start partitioning heritability in a slightly different way. So there's a question by Ayush. How do you know that the low significance SNPs are explaining variation instead of adding random overfitted noise from a bigger number of parameters? Because you're doing this in a cross-validation setting. You're basically learning on a set of individuals and then going off and applying on a different set of individuals. So if it was just simply random noise, you would not be overfitting. Basically, you would not be sort of increasing the predictive power. Ayush, does that answer your question?
Yeah, kind of.
Okay, great. So where it gets interesting is as we start partitioning heritability. What does that mean? That means that instead of looking at the full correlation between phenotypic variation and genetic variation, basically instead of looking at phenotypic correlation between a subset of individuals and genetic correlation between a subset of individuals, we're now going to calculate this genetic correlation between individuals for a subset of SNPs. So you could basically say, if I knew all of the causal SNPs, that's what I would focus on. Or you could say, I'm going to focus on all of the SNPs in the genome. But you could also partition your heritability into different components of heritability and say, well, what about the chromosome 1 component? Or what about all of the SNPs that are active in embryonic stem cells versus the rest component, and so on and so forth. So we can basically start partitioning the GG correlation to be calculated only in a subset of SNPs. So when we do that, we can basically see that if we partition by chromosomes, we basically see that chromosome 1, which is the longest chromosome, captures one of the highest phenotypic variances. And chromosome 2 and 3 and 4 and 5 and 6, as they decrease in length, capture less and less of that phenotypic variance. And some of the smallest chromosomes, 18, 19, 20, 21, 22, are in fact capturing much less phenotypic variance. So what's really fascinating here is that the more SNPs I add, the more I seem to be explaining more heritability, more phenotypic variance. And that's a little puzzling. It basically says that no matter how I cut the genome, that roughly the more genetic space I include, the more heritability I capture. And that sort of moves towards a model where every location of the genome contains some uniformly distributed set of variants that explain more and more heritability. Of course, there are deviations. Chromosome 2 likely contains one or more loci that explains more heritability. Chromosome 7 doesn't. Chromosome 15 probably does as well, and so on and so forth. So we're going to basically extend the model so that chromosomes can explain different proportions of variance. And the intuition is that we're going to add more variance parameters for every partition of SNPs, and every partition will induce a different genetic relationship matrix. So as we start talking about these, E of P as a H equals H squared of E of GG, that genetic relationship matrix can be computed on different subsets of the full set of SNPs. And what we're finding again is that longer chromosomes explain more heritability, suggesting that causal variants are spread almost uniformly across the genome, which is again mind-boggling. And if you look at different traits, dyslipidemia, for example, has a lot of high concentration loci, but also a lot of additional heritability throughout. Hypertension has a smaller number of these really strong loci, and again, many more loci explaining small amounts of heritability. And if you look at schizophrenia, you see almost no screaming signals, but the entire genome capturing roughly the same amount of heritability. So we can basically fit a model with one component per 1 megabase window, and we can bound the cumulative heritability explained to estimate the number of regions. And what we're finding is that most of the genome explains non-zero heritability. Again, pointing towards this, almost complete omnigenic model of disease. So we can also ask, well, can I actually use a Bayesian approach, an empirical Bayesian approach, to select subsets of SNPs based on phenotypic classes that are strongly enriched for SNPs that are associated with a trait? And if I look at type 1 diabetes, I basically see that, IL-2 signaling and measles has, a large number of contributions. If I look at Crohn's disease, I see that, all of these immune pathways are in fact contributing a large amount of that, heritability. Basically, the SNPs are enriched in specific pathways. For rheumatoid arthritis, same thing. For type 2 diabetes, same thing. So then the question is, can we systematically prioritize those SNPs that fall in these phenotypic subclasses in order to boost our power? And the idea is to directly fit the underlying linear model so that there's concentration with these spike priors in these phenotypic classes. And then there's these more flat priors outside these phenotypic classes to basically force many effects to be zero and to regularize the problem. And the goal is to estimate the effect size and the level of sparsity, both within and outside these phenotypically concentrated loci. So we can basically inform with these pathways an empirical prior for these enrichments. Instead of saying I'm going to treat all of the SNPs in the genome with equal probability, I can boost up the prior for SNPs falling in these pathways using an empirical prior that is derived by the enrichment of these pathways. So if some pathways contain more causal variants than the rest of the genome, I can incorporate that into the prior and identify relevant immune signaling pathways which are not found using existing methods, and identify tens of thousands of SNPs that could be affecting those pathways. So again, it's extremely polygenic, but it does still concentrate in specific pathways. So you can basically ask, what are the potential disease architectures that are compatible with the genome-wide association data? And you can actually simulate a disease model. You can simulate realistic loci using the known population and evolutionary parameters. And that's where Agarwala et al. Went. You can simulate disease phenotypes by varying the number of causal loci, the amount of heritability, the prevalence of the disease, and the strength of selection. And you can then perform twin studies, genome-wide association studies, and compare the predicted results to the observed results using this forward simulation. So you can basically design different types of disease architectures and then say, not are they right, but which ones are compatible or incompatible with the data. And you can reject the ones that are incompatible and keep the ones that are not rejected by the data as potential feasible disease architectures. So you could basically say, well, maybe these are my parameters, that basically the sibling risk is this, and basically I'm going to have a polygenic risk score with that, and the specific analysis with a particular chip is going to look like this, and the genome-wide association study is going to look like that, and so on and so forth. And basically, given different parameters of selection of, for example, the coupling to selection of type 2 diabetes could be very strong or very moderate, or just no coupling with selection. And then you can see, are the corresponding data points that we have compatible or incompatible with different models? And for example, if there's a very tight coupling with selection, then that would lead to a sibling risk, which is simply incompatible with the current range of the sibling risk. So we can reject that model. If there's no coupling to selection, then we would expect to have, a large number of genome-wide significant loci, but in fact, we only have 2. So then we can reject that model, and so on and so forth. And in the end, you can maintain the subset of models that are still compatible with the current observations. Okay, so let's see who's with me so far. Great. So we're at 22, 44, 22, 10, 11, and 0. So we can also start incorporating identity by descent in unrelated individuals. The idea here is that for most of genome-wide association studies, we assume that if 2 individuals share the same value of a SNP, that's identity by state. That's basically simply saying, oh, you have a G here and I have a G here, even though we're unrelated to each other. But identity by descent basically means that I have a G here and you have a G here because we actually inherited that exact chromosome from our common ancestor, okay, that we are in fact partly closely related. So you can actually directly capture that using a set of SNPs in a genomic locus to basically look for haplotype sharing. And you can actually build a tree relating all of these individuals. This is the ancestral recombination graph that we had talked about before, and basically ask how are individual haplotypes that are shared contributing to the disease, because those shared haplotypes likely capture additional effects of rare variants that are tagged by these haplotypes. You can also partition SNPs by manual allele frequency or by LD blocks, and then look for assumptions that just simply do not hold in real data. So again, these shared haplotypes can explain more heritability than the tagged SNPs. So the idea is the following. If I find that in my ancestral recombination graphs relating all of the individuals in my study, there's this segment that is not just identity by state, but is identity by descent, namely it was inherited from the same parent. That basically means that I can attribute additional heritability to these segments that would relate these rare variants that are tagged by the common SNPs in those segments. So the shared haplotypes, in fact, do explain more heritability than simply the tagged SNPs. So there's still a discrepancy between, between GWAS heritability and narrow sense heritability. And if 2 individuals share a chromosomal segment, the unobserved variants could also be shared. And then the idea is, let's identify these identified by descent segments, quickly scanning for SNPs and finding stretches of identical alleles. And then we can infer these shared segments that capture these rarer variants more effectively than simply linkage disequilibrium. And you can also partition SNPs by manual allele frequency. And what you find is that the low frequency and the low linkage disequilibrium variants are in fact poorly tagged by observed and imputed variants. So their estimated variance for them needs to be computed separately. And that partitioning appears to explain all of this heritability of height using only common and low frequency variants, which basically suggests that this is simply these, long tail of weak effect variants rather than an additional pool of strong effect variants that we're not capturing from GWAS. So if you start looking at these minor allele frequency and the variance explained by all of them, as you start getting to this very rare part of the frequency spectrum, you don't see any additional variance that is explained. So that suggests that, you know, when you add up all of these together, you know, roughly 10% each, you're, you know, close to this 80% of this heritability of height. Okay, so that suggests that the rare variants are probably not playing a very big effect in height. So again, we can go back and examine all model assumptions. Perhaps the phenotypes might not be Gaussian. Perhaps the GWAS samples are not independent and identically distributed. Perhaps the SNPs are not independent of each other. Perhaps not all SNPs have an effect. Perhaps not all causal SNPs have equal effects. Maybe there are gene-environment interactions. Maybe there are gene-gene interactions. And again, all of that, we're still quite unable to capture for now. All right, so let's see who's with me so far. So we've basically talked about overall heritability. We've talked about polygenic risk scores and how to compute those. And we've talked about ways of distinguishing different types of disease architectures and what the implications are for the number of causal variants and the number of, and their effect size distributions. We're at 50, 30, 20, 0, 0. Okay, so the next topic that I really want you guys to wrap your heads around is this concept of linear mixed models, or LMMs, which have been extremely powerful in statistical genetics. So what is a linear mixed model? So this is a review of our linear algebra lecture. So the idea is that we have n individuals from 1 all the way to n, and we have p SNPs that we're measuring for each individual. So we have, you know, I don't know, 100, maybe 10,000 individuals, and we have 6 million SNPs. And what we're asking is, what is the phenotypic prediction based on the set of genotypes and based on a set of parameters, which are the betas of our effect size model, applied to every one of those SNPs. So basically, the first SNP at position 1 has effect size 1. The second SNP has effect size 2. The third SNP has effect size 3. And so on and so forth. So in matrix notation, we can basically express phenotype Y as a factor of genetic information X. So we're basically multiplying all of the effect sizes for every individual based on their dosage at every genetic location. And that gives us the predicted value for that phenotype. So we're basically asking, you know, what is the deviation between that predicted value and the observed value? And how does that relate with noise, which is Gaussian distributed with some variation? So basically theta is the effect size, which can itself be sampled from a normal prior. So you could basically have a hyperparameter from which you're sampling this, you know, distribution similar to a Dirichlet model. So then the question is, what are we missing in this multivariate model? Well, what we're assuming is that the individuals are IID, are identical, are independent and identically distributed, and that might not be true. So what we could do is add a random effect to account for this unknown. We can basically add this, you know, noise, this additional amount of noise, which is actually driven by these random effects. And then these random effects could be captured by a kinship covariance. And a kinship is basically telling you just how much these individuals are sharing their genetic ancestry. So in GWAS, the most influential and spurious random effects stem from population structure. So then the question is, if I capture that kinship matrix quite well about how much of the genetic variables is shared by any pair of individuals, this actually gives me that additional effect. Okay, so we can basically have now a model where there's a SNP by SNP contribution, and then there's an additional contribution based on the sharing of genetic information between individuals plus that Gaussian distributed noise. Okay, so that epsilon is the same, but now we're adding this additional random effect model. Okay, so why do we need this random effect? Because there's an unknown population structure which can influence simply way too many SNPs, and that phenotypic variation might be due to both population structure and the actual association. So in addition to this association that's flowing through the individual SNPs, we're basically asking what is the association driven by kinship. So we can basically use this a Bayesian approach to account for this random effect mu, where our likelihood model basically is including the additive effects of all of the SNPs and this additional kinship model. And then the Bayesian method would be to address and remove the uncertainty by averaging out this P of Y given all of this, given all the effect sizes multiplied by the dosages. And then you integrate that over all possible kinships, basically. And then this linear mixed model effect is looking for, this, where this new type of noise is now incorporating two components, both this, this zero mean and this IID component, but also a kinship component. So we've basically transformed this problem into sort of combining both the kinship and the random error into these two components. One is the kinship component, which is basically based on the amount of sharing between every pair of individuals. And the other one is this IID error component, which is basically saying what is the deviation for every one individual. So these linear mixed models are basically expressing this phenotypic variation as a function of this heritability, and then these, based on the sharing of the genetic correlation matrix. And then you have this joint model of all the SNPs, which now has been shown to explain more heritability than simply using any kind of subset of SNPs. And under some suitable assumption, this additive component of the phenotypic variation that I can explain by genetics is simply the sum of all of the betas under this infinitesimal assumption where these betas are distributed according to some fraction of the heritability that is captured by every SNP. And you can estimate this additive component without actually estimating the individual betas using residual maximum likelihood, or REML. So what REML does is that it avoids this maximum likelihood fit of parameters, and instead it uses a transformed data so that the nuisance parameters have no effect. And in the various component analysis, this random mixed model, this transformation focuses on the differences and the sums of the variances. And what's really remarkable is that this actually works despite not knowing the causal variance. So for example, with height, the subset of genome-wide significant loci only captures 16% of the phenotypic variation. We would expect, based on all of genetics from kinship and first degree and second degree and third degree relatives, to capture 73%. And if you look now at the entire genome rather than just a subset from GWAS, you can now, using this model, using this linear mixed model, you can actually get to 50%. And then this is driven because we actually don't need to know these estimated betas, because you can actually factor them out using this residual maximum likelihood model. So you can basically generalize this Haseman-Elston regression that we saw previously of this genetic correlation versus phenotypic correlation. And you can basically use the genetic relationship matrix G to capture the identity by state in unrelated individuals. And that's the probability of sharing the same allele at the causal variance. And this is basically known as PCGC regression, which is basically phenotypic correlation by genotypic correlation. The phenotypic correlation is basically taking pairs of individuals and asking how shared are their phenotypic variables. And then GC is genotypic correlation, which is basically asking how shared are the subset of genetic variables. So that's phenotype correlation, genotype correlation regression. So this is just a quick review of these linear mixed models that are basically using this kinship matrix rather than just these error models to basically capture additional genetic contributors that can result from these rare unobserved variants. Okay, so who's with me so far? Lovely. We have 86% at 60 to 80 and 14% at 40 to 60. So let's now dive a little bit more into partitioning heritability. So again, the limitation of heritability is that explaining all of the heritability of complex traits is simply not enough. As the sample size goes to infinity, the question is, will the entire genome be associated with all traits? And our goal is to find biological pathways that are recurrently disrupted by this non-coding variation. So one of the big lessons that emerged from the ENCODE project is that as you start looking at lower and lower thresholds of association, the enrichment of these pathways continues to be high. If you look at, for example, the QRS interval in heart repolarization, so there's different landmarks, P, Q, R, S, etc. This QRS interval in heart repolarization duration is correlated with, heart arrhythmias and all kinds of other phenotypes. But if you look at where are the genetic variants that are associated with QRS localizing, they localize in heart enhancers and heart DNA's accessible regions. And that's true if you look at, 10 to minus 15. That's true if you look at 10 to minus 8, 10 to minus 7, 10 to minus 6, and so on and so forth. And that continues to be true all the way at 10 to the minus 1 or 10 to the minus 2. What's really remarkable here is that the pathways that are associated with disease continue to be enriched very, very far down the heritability, very far down the GWAS association threshold. And you see the same thing with multiple sclerosis, where these immune pathways are very strongly enriched at the top, but also enriched all the way down. And with Crohn's disease, same thing. T helper cells are enriched, at the top, but then continue to be enriched all the way down. So the weakly associated variants overlap accessible chromatin more often than expected by chance, all the way down to the, insignificant thresholds, especially after genome-wide significance testing, about multiple hypothesis testing. And then the same trend is observed in many other predicted regulatory elements, in histone peaks, in chromatin segments, in super enhancer clusters, and so on and so forth. So we can start building this joint model of SNPs and annotations using a penalized stepwise regression to pick relevant annotations. So, this is a model by Joe Pickrell and others in 2014. So we can use an approximate Bayes factor to compute the posterior probability of association by combining the empirical prior, which we estimate from that class, with the likelihood of each SNP in isolation. So the forward steps are adding annotations to the model until they don't explain enough variance, and the backward steps are removing annotations from the fitted model until the variance drops too much. So using this iterative approach, you can basically start asking what are the most, informative priors for every single trait. And you can basically ask, for example, transcription start sites are very strongly enriched. Coding exons are very strongly enriched. Repressed regions are very strongly depleted in multiple cell types, and so on and so forth. So you can build basically this joint model by using approximate Bayes factors to compute the posterior probability of association, and that posterior probability allows you to reprioritize new GWAS loci. So you can basically now look for this posterior probability of association, this PPIA, which now includes many additional ones that are now prioritized based on this weighing by the prior. You can also look at this partitioning of heritability by looking at the subset of SNPs that are localized in specific annotations. So if you do that, you basically see that if you partition your SNPs to only include DNA's hypersensitive sites, then that gives you a dramatically higher fraction of heritability than what you would have expected by chance. If you look at coding regions, the expectation is in this dashed line. And indeed, coding regions are dramatically enriched. UTRs are also dramatically enriched. Promoter regions are also dramatically enriched. By contrast, intergenic regions, you would expect them to cover this much based on just a fraction of the cover of the genome that they cover, but in fact, they cover much, much less. They're actually strongly depleted for heritability, and so are intronic regions. By contrast, you see this dramatic increase for all of these functional annotations. What does that tell us? That basically tells us that if we compute the heritability by focusing on the subset of SNPs to be thought of as causal that are sitting in these different annotations, then we can explain basically 80% of the heritability on average using these DNase hypersensitive sites, these DNA accessible regions, which are very strongly enriched for genetic variants associated with TDs, and they're very strongly predictive of gene regulatory elements. So accessible chromatin explains more heritability, a lot more than what you expect. And if you combine DNase hypersensitive sites in more than 100 cell types, then 70% of the genome is accessible in some cell type, but only 60% in multiple cell types. That implies that non-coding SNPs, in fact, explain more variants per SNP than even coding SNPs, that this enrichment is quite remarkable. So I use by an annotation here, we mean different labels for that sequence. For example, if it's annotated to be a coding region, a UTR region, a promoter region, and so on and so forth, instead of just looking at the genome in a flat, unlabeled way, you start labeling parts of the genome, and these are the annotations. So everybody with me on this concept of partitioning heritability? Yes? Awesome. So the next challenge is, how do we correct for the effect of linkage disequilibrium on these regions? So that's what LD score regression tries to do. So here's the intuition behind LD score regression. The intuition is that causal variants are drawn uniformly at random from the genome. And if they're drawn like that, they're more likely to come from larger LD blocks. Why is that? Because if I basically have multiple tagged SNPs that are all co-inherited, then the combined effect of all of those SNPs is in fact continuing to increase the association of any one of those variants. And basically, if there's a causal variant here, I will capture it by many additional tag variants, and I will boost the power of being able to identify it. So I can actually do a linear regression of the summary statistics versus the LD score, which is summing up the amount of LD, the amount of linkage disequilibrium that every SNP has with its neighbors. And that can basically give us heritability without actually having access to the individual level genotype matrix. Because I can basically look at the LD score matrix, which is basically capturing the amount of sharing between any pair of SNPs in my cohort without actually needing the individual level genotype. I can just simply infer based on the correlation between my entire cohort for that SNP and the entire cohort in that SNP. So I can look at that correlation, and that's simply given by the R-squared matrix. So I can use this LD score matrix to effectively get at the underlying regression without actually having access to the genotypes. So what LD score regression can do is now estimate heritability from simply summary data. So you can build a multivariate model for phenotypic variation, where the phenotypic variable for individual I, yi, is a function of the full set of effect sizes estimated and evaluated based on the dosage of reference of alternate alleles for every SNP, and then a non-genetic contributor. And assuming that the expectation of these genetic loadings are in fact 0, and this variance is 1, then the heritability is given by V of XB, which is the sum of x squared b squared for every one of those loci, and it's basically the sum of all of these b squares. So basically that heritability is effectively the sum of the effect sizes. And by partitioning, by restricting on a particular set, then that heritability partition on that set is simply the sum over just those subsets multiplied by that effect size. So what LD score regression does is that it estimates the heritability from the summary data. So using this multivariate model, I can basically infer the summary statistics using this chi-square test statistic for all of the SNPs, and this LD matrix or correlation between SNP and SNP and SNP. So that R squared between any pair of SNPs, that's basically the LD score or the LD matrix, or simply the correlation between any pair of SNPs. And I just need to know this genetic correlation matrix for my individuals. I don't need to actually know the individual genotypes for my set of individuals. So basically I can do a heritability by partitioning, by restricting to the set as the sum of all this, and then this overall heritability is that. So then the idea is, let's reverse engineer the summary data to find the multivariate parameters. So if this is the R squared between SNP and SNP, I can basically compute these betas by correcting for this R squared. And then I can look for this univariate chi-square model for GWAS, where you basically have this, distribution according to these betas and this R squared distribution and this modified E, which also includes, these additional parameters. So then the per SNP variance in heritability is, which is basically this heritability is this, under the assumption that these betas are distributed around 0, this E of beta squared can basically be plugged in, right in here, which is simply the variance of these betas, which is simply this sum of over this particular variable. So then that gives you this estimate of the total effect of all of these being basically partitioned into these components, where you basically have this, error of this residual. You have this R squared, and then you have these corrected betas for every one of those locations, which is basically the variance of the betas at every one of those locations. So then you can regress on the chi-square statistics of these LD scores. You can basically take this LD score of every pair of SNPs, this LD score of the set of SNPs, all within the same set of annotations. So this is the LD score between every SNP J and all of the other SNPs K, which are sitting within the same annotation that you're looking at. And then you can remove all of the unwanted double counting of annotation enrichments by LD by knowing the R squared of all of the SNPs in your sample. You can basically correct within each annotation for that LD between all of the SNPs in the same annotation. So you can basically do this regression to estimate this residual tau that you can basically now correct for every one of these annotations. So if you have P SNPs and P observations, you basically have these, lambda, which is this LD score between every single SNP in that annotation and your SNP of interest. And then that entire correlation matrix is basically allowing you to now partition this heritability score within each annotation class. And then, get at that R squared matrix, this correlation matrix, this LD matrix, and then the corrected residual for the effect size for that functional class. So you can basically have a total sum per functional class and LD score correction for all of the correlations between pairs of SNPs within that class, between your SNP J of interest and all of the other SNPs K within that class, and then that residual. So using this stratified LD score regression, you can now start partitioning the heritability of complex traits using just the GWAS summary statistics. So you don't need the kinship matrix. You don't need the gene, sorry, all you need is the G by G correlation for every genetic variant and every other genetic variant within the class. And you can use that to now start partitioning the entire heritability without actually knowing the individual level genotypes for every one of your individuals. And when you do that, you can basically see that the heritability partitioning of schizophrenia across these different functional classes, so basically these are the classes that we're looking at, across these different functional annotations, concentrates specifically in enhancers active in the central nervous system. For bipolar disorder, same thing. For height, it concentrates in connective bone and cardiovascular annotations. For Crohn's disease and rheumatoid arthritis, it concentrates in immune and hematopoietic enhancer annotations. For fasting glucose, it concentrates in adrenal and pancreas. For BMI, it concentrates in both brain and adrenal and pancreas. And for education or smoking, you see this concentration again in the central nervous system. And for HDL cholesterol, you see this concentration in the liver. And for age at menarche, which is basically connected both with adrenal and with central nervous system. So that basically tells you that this particular method for partitioning heritability can actually be very powerful in splitting up these functional categories and elucidating the subsets of the genome that actually contain the most heritability. So let's see who's following so far. Okay. So we're at 2050, 2010, 0. All right, so we basically talked about heritability, polygenic risk scores, disease architecture, linear mixed models, partitioning heritability, LD score regression as one way of partitioning heritability. Let's now talk a little bit more about polygenicity leading up to this omnigenic model. So the question is, how far down the SNP list does the enrichment go? So we can use the functional enrichments to gain insights into genetic architecture. And then the idea is that as we consider more and more SNPs beyond the genome-wide significance, the relevant regulatory regions will be disrupted more often than irrelevant ones. So we can basically ask if this annotation is relevant. We can see if it is prioritized early in the list of all SNPs, and then they all end up back down to where you expect them to be. So basically, after you include the entire genome, every region will be enriched according to its overall representation because you've now included all of the SNPs. But the question is, as I sort the SNPs by their p-value, are particular annotations very heavily enriched early on? And then how far down the list do I continue seeing enrichment? And what's really remarkable, as I mentioned earlier, is that there's very long tails of enrichment for a large number of diseases. That basically, if you look at immune disorders, they continue going down. Even for Alzheimer's disease, you can see this immune association. For schizophrenia, you see this both immune and neuronal association. For coronary artery disease, you see these sort of muscle associations. And for type 2 diabetes, you see this pancreatic islet. And in each case, you see that this association goes down thousands and thousands of loci. And these are looking at specifically independent loci by p-value. So that suggests that there are really thousands of functionally relevant SNPs and thousands of individual independent functionally relevant loci concentrating in the same pathways. So you can now start carrying out the systems genomics of this. You can start grouping all these loci according to modules, for example. So you can take the 127 epigenomic annotations of roadmap and then start grouping them into modules of coordinated activity, and then start looking at how are these modules correlated with specific functional annotations. And the challenge, of course, is that the annotations learned in one cell type at a time can't account for the sharing of elements across cell types. So we need to use some kind of clustering to define modules of enhancer activity that are cutting across multiple tissues. For example, this module here, 69, is associated with both T cell and B cell function. This other module here, 77, is associated with, function across all of the cell types. This other module here is associated with primarily brain function, but also some additional function in internal organs and so on and so forth. So as you start looking at these modules, you can see that the functional enrichment now highlights the importance of both constitutive and linear specific enhancers. So for each of those disorders, you can see that the constitutive enhancers as a class are very strongly enriched, indicating this sort of common component to many traits. We can now use these to go from enhancers to the target genes and to their pathways. For each trait, we can basically ask what are the genes downstream of these enhancers and what are the pathways that these genes are falling into. And we can link enhancers to the downstream target genes and see that the target genes are enriched in known disease pathways that were, but through previously unknown mechanisms. So you can reveal this broad similarity at the pathway level between classes of diseases with signaling, underlying autoimmune disorders, etc., but also specific pathways that are important to each disease. And that can basically potentially implicate novel genes in enriched pathways. And then you can even go to the upstream regulators. You can ask, from the set of genes that are enriched in coronary artery disease, can I find that there's a set of co-regulated modules that are all downstream of the same upstream regulator? So you can basically start looking at these upstream regulators using sequence-based enrichment without actually considering the GWAS, and then find master regulators that are recurrently disrupted by subthreshold SNPs. And you find many disease-specific regulators and some interesting shared regulators. And you can now start building these networks of regulator to gene, and then asking how are the target genes of these regulators consistently linked to the same set of genetic variants associated with the same phenotype. And then these upstream regulators can add the cell type specificity. So you can basically ask, what are the predicted master regulators that are found in predictive constitutive enhancers rather than cell type specific regulators? And then although the enhancers might be constitutively marked, the expression of the upstream regulators is still cell type specific. So that gives us additional insights in the transcriptional regulatory regulation needed to interpret non-coding disease associations and start getting at who are the regulators and what are the cell types in which they're acting and what are the traits in which they're acting and how are these traits supported by the specific cell types. So what this polygenic model is telling us is that there's a large number of contributors to the trait that are cutting across both disease-specific and disease-shared models. And the last topic that I want to cover today is this omnigenic model of heritability, which is basically trying to get at directly this. And a huge enrichment basically means that it's more than what you would expect if these variants were uniformly distributed in the genome. So this omnigenic model of heritability is basically saying that if I start looking at these cross-cutting annotations, not of a particular regulator, but of models that are telling us about active enhancers or polycomb repressed enhancers or EQTLs, what we're seeing is that compared to the expected p-value, the observed p-value is dramatically enriched. And that enrichment happens very, very early on in this p-value distribution, not just at 10 to the minus 8, but very, very early on. And the estimated fraction of SNPs that associated with, for example, height as a function of LD is continuing to be dramatically enriched. And even if you look at this sorting based on the p-value not in a log space anymore, but now in a linear space, you see that this enrichment is not just simply stopping here, but is continuing almost all the way to 1. So yes, the distribution is extremely broad, but it is consistently above 0. So that basically tells you that the median effect of all SNPs is actually 0.1 millimeters, that nearly all SNPs in the genome are in fact contributing to height with a tiny little effect size, but all of them are. So the question is every SNP in the genome associated with height? And the answer is that there is in fact more heritability to be captured in these broad functional classes. That if you basically look at what are active in the top cell type and which ones are uniquely active or specific or broad, that in fact these broadly active classes are in fact capturing a lot of heritability. And if you look at the SNPs that are broadly that are near these broadly enriched genes, they explain more schizophrenia heritability than those near brain biased genes. Basically, this total heritability enrichment is certainly higher for frontal cortex, but if you look at the total heritability explained, you actually have more heritability explained by broadly expressed genes than by frontal cortex genes. So that leads to this very strange conundrum, which is that nearly every single gene ontology category is consistently enriched in these disease-associated genes. That if you look at, for Crohn's disease, protein binding as a functional GO category, sequence-specific DNA binding, immune response, inflammatory response, all of these are, above the expectation. And of course, just to compensate, some of them have to be below the expectation, such as ion channel activity and so on and so forth. But ion channel activity is in fact enriched in schizophrenia, along with a bunch of other things like protein binding and sequence-specific DNA binding and so on and so forth. So what's really weird is that across many very different disorders, the same set of functional classes continue to be systematically enriched. And the way that Jonathan Pritchard and Alan Boyle and others have basically sought to explain this is that there's a set of core genes that are associated with a specific disorder. These are, for example, the brain genes for schizophrenia. And then there's a set of peripheral genes which are associated with protein binding or RNA metabolism or, metabolic processes more broadly and so on and so forth, which are surrounding these pathways and affecting these pathways through their interactions, but are together capturing a lot more heritability than the core pathways by themselves. And that basically GWAS has done a very good job in capturing these core set of genes very early on. But as you go further and further from those, that basically larger and larger GWAS studies are in fact showing that there is in fact non-random, enriched heritability continuing to cluster well outside this core network. So that's the current view of systems genetics at the systems level, or at the genome-wide level. We've basically studied heritability and shown that it is very widely distributed across almost every megabase block in the genome, that these polygenic risk scores allow us to capture more and more accurate models of phenotype by capturing not just the genome-wide significant SNPs, but a huge number of additional weak effect SNPs in the genome. That disease architecture is usually consistent with these very large numbers of weak effect SNPs contributing together. And linear mixed models are one way to capture these effects, even when we don't know the individual betas with sufficient confidence. And we can start partitioning heritability across functional categories by subdividing the genome, either by chromosome or by functional category, or by the set of enhancers that are active in any particular subset of tissues or modules of combinations of tissues. And LD score regression is a particular way to correct for the impact of neighboring SNPs and to exploit that impact of neighboring SNPs through these R-squared correlation matrix in any subsets of functional categories. And what that tells us about the heritability through the shared kinship of every one of these categories without actually having to have access to the individual genotypes of every person participating in the study. And then how this polygenicity is in fact pointing to these global regulators that are underlying these traits and these thousands of genetic loci that are contributing, and how these are in fact concentrating in specific pathways that are core to the particular trait, but that those pathways, even though they're the most enriched, are in fact still capturing a minority of the heritability, and that the majority of heritability is in fact captured in this periphery of this network of regulators and factors and cofactors and interactors that are contributing to the overall heritability of the trait, but not directly, but instead indirectly through the peripheral genes. So let's see who's with me so far on this lecture and its messages. Very cool. So we have 20, 20, 80. Oh, let's change to 30, 15, 60. And then lastly, who feels that they've learned something today? Very cool. So we're at roughly 50-50 between, yes, this was great, and yes, this was interesting. And, so basically 5 and 4 are dominating here. So we have 43, 57, 0, 0, 0. All right. So thanks, everyone. And then looking forward to seeing you next time when we're entering the new module on evolution. Thanks, everyone. Bye-bye.